Welcome to this webinar on crystallization process development, optimization, and scale-up. My name is Nima Yazdanpana. I'm a principal consultant at Procigens, and I'm going to show you how we scale up from lab to manufacturing by digital twins and mechanistic modeling. This video is a recording of a previous webinar we recently had for Technobees. I put a link to the main page here, and I'm also going to add the link below this video. Feel free to check their website and get more information about about our work and also the technology they have with the Crystal 16 and the Crystalline. A quick word on Procigens, who we are, what we do. Procigens, our name stands for Process Intelligence. We are a consultancy firm helping pharma, biopharma, and fine chemical industries to scale up from lab to manufacturing and reduce time to market, increasing R&D efficiency and reducing cost and waste. So we have had many different activities in different areas, different scales, different type of product, drug substance, drug product, biologics, devices, ranges from like a spray drying, hydrogenation, flow chemistry, techno-economic analysis. But for the purpose of this webinar, I'm just going to focus on crystallization. If you are interested to learn more about how we help companies to scale up and optimize their equipment and the processes, feel free to reach out to us at info@procigens.com. Here you can see some saving metrics and saving calculation we have had for the couple of our projects we have had with our clients. And so we check how much they save on the cost or the time, improve the quality and the yield. You can see instead of spending time and material and expensive resources in the lab and manufacturing scale, how we can use modeling and simulation and in-silico tools to reduce the time and the cost and bring a product to market cheaper and faster and better and higher quality. Talking about the scale-up challenges, what would be the cost and time and resources here? As an example, we're discussing today on a crystallization. So crystallization, you can see, for example, here with the EasyMax, something maybe 100 milliliter. It takes like 1 day or a few hours to run one experiment for crystallization, depending on the type of the crystallization process. For example, batch cooling crystallization here. Operation cost is manageable, it's not that expensive, so we are dealing with 100 milliliter of the solvents and a small amount of material we have to use. Material cost and availability as I mentioned is still manageable, and we have multiple options on using different vials, different glasswares, different designs of the reactors, or baffles or impellers. However, when we go to manufacturing, now the scale is very large. It's like 100,000 liter, 200,000 liter crystallizer or reactor. That takes longer time to process, 2-3 days, because the cooling rate is different, and preparation and the cleaning and running all the experiment end to end, it's a longer time. Now we are dealing with 100,000 liter solvents, so the operation cost and the material cost become very expensive, and also the solute is a precious material that we're trying to recover. So it's kind of like impossible to run a DOE on the manufacturing scale and trying different combination of the process parameters. Also, most of the time when we go to manufacturing, would be a brownfield project, and equipment exists. We have limited degree of freedom of changing the design of the equipment, such as type of baffle, locations, or the sizes, or volume to height ratio of the vessel, and how it's going to be impacting all the process parameter that would also impact the crystallization process. So we don't have much degree of freedom on the equipment design. However, when we scale up, like by 1,000 times larger or like 100 times larger, the parameters of the system are non-linear. In this case, so is a quick calculation. You can see the volume from like a 10-liter reactor crystallizer. We're going to like 100,000 liter. Now the volume is 10,000 times larger. However, most of the time the RPM or the agitation rate would be the same, even lower, because at a larger scale we cannot go. Normally we can not go above like 250 RPM or maybe 150, depending on the size and the height of the crystallizer, because of the vibration and the mechanical issues the electromotor would cause us. However, the cooling rate would be like significantly lower because of the heat transfer area ratio to the bulk of the solution. So it's gonna be like 100. So you can see the volume is like 10,000 times larger, but the cooling rate is like 100 smaller. And finding all these nonlinearities is very important to have the knowledge about what parameters would impacting the process, the yield, the quality of the material, and that would be something impossible to do to do the experiment and find the best condition at the first time. What we use instead at Procygens, and we're helping our clients to do that. So we're using the digital twin, so to speak, is a knowledge map, is understanding about all the parameters, all the underlying physics and the thermodynamics and the kinetics, and we go on a different stages, different steps. So the first step, or what we call like the ideation here, so we can run a multiple simulation, kind of like a parametric study, changing temperature, pressure, flow rate, RPM, many different parameters, and find the best condition that would work on based on the simulation. However, all this simulation could be wrong. Yet the results, the graphs would be colorful and very shiny, but the values could be completely wrong. So we have to do validation and verification every time that we're developing a model. It doesn't mean that you have to validate every single data point from the model. So we have our in-house, we have some SOPs to how to do the validation and verification for the model, and maybe with one or two data points. Anyway, so that would be something manageable in the lab with one small experiment, short experiment. We could do the validation to check the model results, and when we have confidence on the simulation data, simulation performance, so we can use for scaling up to a very large scale. And then when using this validated model from the lab and use it for the larger scale, now we can run again parametric assay to find the best combination of different parameters that would gives us best performance at the larger scale. So kind of eliminating the DOE for the larger scale, or performing a virtual DOE with these kind of simulation tools. The most important part of this knowledge management is the collaboration, is communication. So the knowledge that be generated from the chemistry stage, solubility, for example, that would be useful for finding the solid equilibrium, that develop the knowledge for the crystallization, for the supersaturation ratio detection, and then that would be communicated from the chemists to engineers to the simulation persons, and again from lab scale to manufacturing, and then maybe the internal parties or external, such as the CMO. So we all have to talk together, work together, and put all this knowledge together to develop that kind of knowledge-based process product development. The digital twin has multiple steps. The core part of that is the chemistry, the biology, the solubility, in this case for crystallization. And then based upon that, we built up different levels depending on the application. For example, what we call like equipment model, it would be for one unit operation, in this case crystallizer, what would be the impact of the shear rate or the cooling rate, or how is the energy dissipation through the bulk. All of these things could be simulated, and then we would have like a virtual model that could be or like a digital twin of our actual process, and then adding up the layer on top of each other. So everything based upon still like the core, the solubility or the chemistry. Then we can have to like the process model, right? At the process model stage, we here we have like a one-way communication with the monitoring system, with the SCADA system. We're getting the data from the plants, from the equipment, that could be used for parameter estimation, kind of dynamic alignments, and you know, the model updates. Then at the topest level, it could be like a two-way communication, that the models, the simulation also would talk to the SCADA system, to the control system, and can change the process by the models, which is called like a model predictive control or advanced process control. So all these called digital twin in literature, and especially recently is being discussed by many companies. However, the topic is not new. So around 20 years ago is more becoming into the processing industry, and before that it was in, like, for example, flight simulator for training the pilots before getting to an airplane. So we can make different levels, but different accuracy, different expenses, different time, and all depend on the goal of the project, right? Are we going to put all the resources to, you know, have a model predictive control? Then we need to have like very extensive digital twin, or is it just for like equipment optimization? That would be just to do the second level. I mentioned the base knowledge is the fundamental information that we have to develop. For example, chemistry or the solubility, and in this case, you know, the solubility can be measured by the Crystal 16, which has you know, 16 experiment or 16 data point per experiment. Solid liquid equilibrium or the solubility is independent of the scale, is more like a thermodynamic dependent. If we know the composition of the solvent, or if we are changing the composition of the solvent, or if we know the temperature, or if we are changing the temperature. So that's something that we could use for you know, developing the solubility curve based on the cooling or like anti-solvent. Metastable zone width, however, is kinetic dependent and variable. It depends on the cooling rate, it depends on the mixing rate, all the kinetic energy that we are going to add to the system. So it's not something scale dependent. There are some other methods for solubility measurements or prediction. For example, simulation with some you know, quantum chemistry, or some you know, thermodynamic models like an RTL uniquark. But again, we need to have some validation for those kind of simulation for the solubility. It still can be costly and expensive. Instead, if you have experiment, if you have like a Crystal 16, or even like you know, gravimetry by like a filter and some balances and an oven, so you could do the measurement directly in the lab, which would be cheaper and faster at this stage. So the solubility knowledge is developed, and now we know how our molecule, our solute, works at different temperature, if this is a cooling crystallization, or if we need to develop a phase diagram with different concentration of the solvents or anti-solvents, and having the phase diagram for that, or reactive crystallization, which would be a combination of both. We have a degree of freedom at a fixed temperature, or changing the solvent composition, which in this case would be the product of the chemistry of the reaction would be insoluble or had less solubility in that solvent system, and then we're gonna precipitate it out as in form of salt or crystals. Going to very over some quick equations, and some not going too deep into the details. First, let me introduce some of the references I use for this presentation. So we are familiar with the basics of the crystallization and the fundamental models, and I encourage you to refer to the textbooks and the basic knowledge around the crystallization, about the solubility, about the processes. And especially I'm using these two books, the Handbook of Industrial Crystallization by Alan Meyerson, who was my PI at MIT, and also the Mersman, which is you know, everyone knows that. Nucleation, crystal growth, and the population balance. These are the three steps that we do the model development for crystallization. There are different equations, different parameters, different methods for modeling these steps. Again, I'm not going to the detail, but for example, for nucleation, I'm showing you one example here, one of the model that could be used, and like to draw your attention to the parameters that I highlighted here, or draw a blue circle around. The C minus C sat, which means the supersaturation ratio, and the end is the exponent is also the rate of changing that the supersaturation ratio, which would impact the nucleation. That is something scale dependent. The tip speed of the of the impeller, so the amount of energy we are putting into the crystallizer by the shear, or by kind of velocity of the tip speed. That's also another thing that is very in the scale dependent, that would impact our nucleation. Magma density, if we have some heterogeneity into the crystallizer, some slurry distribution, that also would be scale dependent, and I'm going to show you in the case studies that also something need to be considered at the new scale. Crystal growth, again, the different type of equation we could use, and we have the tip speed and the supersaturation ratio again here, which these two are scale dependent. There are different also method to model the crystal growth, and which gonna end up to the same concept, same kind of methodology, but they might have different kind of notations here. If the growth rate is at the unknown, so then the reaction rate it would be limited to the diffusion rate. Then we can use more like a Sherwood type relationship. That then I'm showing you the equation here. Then the solid surface concentration can be eliminated using the definition of the efficiency, and it's gonna be like more kind of simplified method that could be used for the crystal growth. Population balance is another useful piece of information, or the tool that we have. However, population balance is a hybrid model. The parameters need to be estimated by the experimental data. So kind of fitting the growth rate or the number of the crystals based on the data that we are collecting from the crystallizer by some PAT tools, for example, FBRM, which measure the chord length, or Blaze metrics that they measure the particle size. Then if we go to larger scale, to a new scale, or changing cooling rate or agitation, we have to redo our population balance. It's a useful tool, but for a scale up is not that much solid tool, so we have to redo it again and get the population balance at the new scale. So there are two type of population balance. So 1D population balance, which gets the overall growth, and also 2D, which I'm going to show you in one of the case studies, that you can predict the change of the crystal in order of width or length, or two different dimensions. Some scale dependent phenomena in a crystallization process, as I mentioned, heat transfer, which would impact the supersaturation formation rate. If the jacket cools down, and now we have a reactor with like 3-meter diameter, how long it takes to cool down the whole bulk, right? And how long it takes to get to the supersaturation that we need enough driving force to drive our crystals out. Mass transfer layer, again, depending on like a mixing and the boundary layer at the interface of the crystal growth is very important. Any local supersaturation is forming, especially for reactive or anti-solvent crystallization. So this would be like significantly scale dependent. The energy in terms of shear, tip speed, or other phenomena that would cause the particle size distribution changes, such as breakage of the crystals, attrition, and impact of like particle or particle impeller or particle wall attrition. That would all depending on the energy and also the scale dependent. We mentioned about the metastable zone width, which is kinetic dependent, scale dependent, It depends on the mixing rate and the intensity of the mixing, and the supersaturation formation rate, which I mentioned before, is the heat transfer rate or the cool down rate, how many degree Celsius per second or per minute we are going to cool our system, and that would change our metastable zone width. I'm using some equation from this literature, and you can refer to this review paper published last year. They had a kind of extensive review of different equations and models for metastable zone width prediction, which is actually from like a Mersman. As I mentioned, this is a review paper. But in this case, you can see the metastable zone width depending on the mixing. If we change the mixing rate, if we have a very rigorous mixing, let's say at 400 RPM versus very gentle mixing, maybe like 50 RPM, then that metastable zone width would be different, right? The location would be different. Sorry, the metastable zone line would be at a different location, which gives us the different metastable zone width. Similarly, for the cooling rates, here is a paper published like a few years ago, 10 years ago, and this is one example of many papers that they use different cooling rates, and by that different cooling rate, by that kind of ramps that go up and down, so you can see the metastable zone width and the slope, that would be at a different location of the, for example, that kind of like a kinetic dependent metastable zone line. Here in this example, we are showing two simulations for the lab scale and large scale. The bottom one is 100 milliliter easy max scale, and the top one is 10,000 liter reactor that we had for a client. For both cases, we are starting at the 50 degrees Celsius, the starting temperature of the bulk solution. For both cases, the bulk start was 50 degree. The jacket temperature is 10 degree, so we try to cool down this solution to the 10 degree, which is our like crystallization temperature, as soon as possible. However, because the top one, the manufacturing scale one, is very large, the bulk or the surface area of the heat transfer or the heat transfer surface ratio to the bulk ratio is very small. You can see the lab scale takes to get to the equilibrium temperature of 10 degrees C at 300 seconds, which is like 5 minutes, and the large guy takes 30 minutes to cool down. This also the RPM of the mixer is a significantly important factor. The RPM gives us the convective heat transfer. So rigorous mixing gives us better heat transfer, and we would have, like you can see here, comparing the 100 RPM and the 200 RPM, be getting the faster cool down at the larger scale. Similar trend for the smaller scale also, but here the green line is at the 400 RPM. Something I would like to show, if I can rewind back the video, maybe here. I mentioned this line, the right side is the wall, is the jacket temperature, and here is the bulk, right? And you can see the temperature difference from at that kind of front, at that close to the wall, somewhere here, is at this time is like 15 degree, 20 degree difference, right? And that may cause encrustation. However, in the larger scale, we don't see that kind of significant difference at like any time. Maybe here would be the maximum. Here, that kind of temperature difference from the wall to the bulk, that boundary layer is like maybe 5 degree, right? So we might see more encrustation or the scale formation in the lab scale, because that the heat transfer is super fast, and we have this boundary layer that has significant temperature difference. Now, if we have a look at the cooling rate when we're changing the RPM, as I mentioned, in the larger scale, so we get to the cold temperature at 30 minutes for 100 RPM, and at 20 minutes for the 200 RPM, because we are changing the heat transfer coefficient, the convective heat transfer coefficient. So, but the rate of the cooling also changes the supersaturation ratio formation rate. Here, if I could draw a line for like a kind of, let's say, assume the temperature of the solubility, let's say 20 degree. somewhere here. And you can see the slope when we changing um, the RPM or the cooling rate is significantly different, right? So this slope, like the theta. So at the faster RPM, so if we make supersaturation ratio faster, that would change the, for example, the exponent for the, the crystal growth or the nucleation, as you, if you remember from the, the equations we had for the crystal growth rate. So now that faster supersaturation ratio formation, it increases the nucleation rate and the growth rate. Also here we have the higher RPM, which increases the supersaturation, the tip speed rate, right? The tau. This is more significant when we scale up from the lab scale 100 milliliter to 1500 liter, and you can see the slope is significantly larger. If I use the same analogy, let's say at a 20 degree, now this slope is kind of shallower, and this is like very steep, like almost 90 degree. And that is the scale dependent phenomena that we have from the lab scale to large scale manufacturing. At the same RPM, same temperature differences, but we get to the higher supersaturation ratio formation rate. Now we get to this conclusion that why a lab developed recipe should be valid for the large scale manufacturing, right? So we see the significant difference between the smaller scale in the lab at the development stage, and now when we're going to manufacturing. So let's also have a quick touch on impurity form inclusion into the crystal lattice. So the impurity inclusion comes from like two different mechanisms, or I would say three. So we would have the distribution coefficient as a kind of like a matrix, or kind of like a definition that how much of the impurity gonna incorporate into the crystal lattice, right? So the equilibrium distribution coefficient, it depends on the thermodynamics, it depends on the solubility, it depends on the similarity of the molecule, the host molecule and the impurity together. For example, you can refer to two of my publications back in 2016 and 17 when we compared paracetamol and metacetamol, which are very, very similar. They have similar structures, and the propensity of equilibrium distribution coefficient, or the propensity of the incorporating the impurities into the crystal for metacetamol paracetamol system would be significantly larger. But if you have significant difference between the molecular structure, for example, in this case, you can see the difference between the phenofibric acid and phenofibrate. And now we would have less impurity inclusion based on just the thermodynamics, just based on the solubility and the similarity of the structures. But that is more on like equilibrium conditions, and that is, kind of fixed. At the higher growth rate, at when we are growing crystal super fast, so there are other mechanisms also getting involved, what we call like the effective distribution coefficient. And you can see in the equation here that the effective distribution coefficient has three different components. The first one is the equilibrium section. So here is a relative concentration of the impurity at the solution, in the solution. How fast we are forming the crystals. If we grow super fast, so we may end up with some solvent pockets, some solvent inclusion. And that because the solvent is kind of entrapping from the boundary layer, is very rich in the impurity. And the other kind of mechanism is the growth rate. When we grow very fast, so the impurity is getting into the crystal lattice very at a higher rate. So when we're increasing the crystal growth rate, when we're changing from the lab scale to manufacturing, also the impurity or the quality of the material would be significantly different. So kind of quick mentioning the equipment design setup and orientation impact. And so if we have different RPM or different location of the probe or different setup for the equipments, how would work in terms of the suspension and the shear distribution and the eddy diffusivity and all of those kind of stuff that's going to impact our breakage, growth rates, nucleation, tip speed, all of that. For example, in this case, we can see this is a smaller scale, is like EasyMax scale, 100 milliliter, and we have the probe at different locations. In this slide, you see the low location of the probe. I mean, so the distance between the end of the probe tip to the impeller is kind of a small. And the right graph shows the distribution from the top to bottom. The top here is on the surface of the solution, and the bottom is the bottom of the flask, right? So we have different colors. The blue is for the fine material like D10, green is for the D50, and the purple is for the large materials or D90. And we can see when the probe is at low location at this RPM, so we have this kind of distribution. When we compare it to higher probe location, so when this distance from the probe tip to the impeller is wider, now we have the kind of, so to speak, baffle impact of the impeller at the which is not that much significant in the solution. Now we can see we have more kind of sedimentation, or we have more number of the larger crystals, the D90 on the bottom of the crystallizer, right? Just by changing the probe location, now it's going to have a significant impact on the suspension density, the kind of axial distribution, and the profile of the sizes from the surface of the crystallizer to the bottom of the flasks. If compare side by side, when we have the same RPM, different probe location, so these systems are like exactly the same RPM, just different locations. Also, same number of the crystals, same number of the particles. Just for the visualization, these dots here in the graphs are not like the actual particles. This is like the statistical distribution, or like what is the chance or the frequency of having the crystals at each location at each time. And you can see when the probe is lower, so we have very nice distribution, so all the particles are everywhere. When we have the probe higher, so there was some like vortex formation, and we would have more probability of having more crystals on the bottom, right? And when we do some like a quantification, you can see this the difference between the bars. Now if we change the RPM at the same location of the probe, here we call it like the medium or like the base location, so somewhere in the middle, comparing at 200 RPM, and then next slide is 400. Just for the sake of time, I just go to the side-by-side comparison. Same location, same number of the crystals, same sizes, same shapes, but now at different RPM, you can see at the higher RPM, so we have more larger particles at the bottom. This sounds like kind of counterintuitive, that if we want to have higher suspension, you want to increase the RPM, so to get them kind of like the fluidize of increasing the drag force. But in this case, so it might be different for other cases, but in this case we can see there is some like a dead zones on the bottom of the flask, just underneath of the impeller, and that's all like the shear distribution that causes these dead zones, and now we have more number of the large particles at the bottom. Now this impeller, because it's just running through this kind of like a thick slurry of large particles, also act as a crusher, so it's crushing the particles. And what happens now? Creating fines, seed generation, secondary nucleation, all of those things could go wrong. On top of that, at the higher RPM, now we have higher tip speed. That also would change the crystal growth rate. So we would have different seeding, different nucleation, and also the different growth rate because of the high RPM that cause different heat transfer, different cooling, and also different tip speed. So this is kind of like a demo. I was walking through an expo, a trade show, and I saw this glass they had there from Chem Glass in their booth, and they had this demo for that was just for the glassware, and I saw the mixing, and I got a video to share with you. I can stop somewhere. Yeah, let's stop here. So you can see this. This is like a two impellers. One on top is a pitch blade. The bottom one is the wacky kind of half moon, and at a very high RPM. So we have this kind of like a vortex formation. So we think that the mixing should be very good, right? But here they use this kind of like a glass beads or plastic beads for the demonstration. And you can see we have more kind of distribution of the particles on the bottom. So on the top, almost no particles, and on the bottom, so we have a very kind of high concentration of the slurry, right? So it would be more prominent at the end somewhere here. What would be the impact of the particle shape? Here we are comparing three different cases. So we did the CFD analysis again for these cases is a CFD-DM. So we have three type of the crystals, let's say three different polymorphs. Form 1 is like 1-to-1 aspect ratio, somehow cubical. Form 2 is more like needle shape, and form 3 is like 2D kind of flaky, right? Now when we have these kind of very flaky plate shape crystals, so it has different drag force and different buoyancy. It's more like kind of like a fluidization, right? So when we have a higher surface area, so the crystals they prefer to kind of flotate or the bounce around for longer time. So again, same RPM, same shape of the vessel, same number of the crystals, but for the different polymorphs, we can see we have more kind of uniform distribution for this kind of like a flaky crystals, these 2D shape plates, comparing to this on the left side, this kind of like a 1-to-1 aspect ratio kind of cubical. Now again, getting back to the nucleation or the attrition, now when we are hitting these crystals together, so it's going to be like abrasion of the particle attritions, and so there's a propensity of forming more fine crystals and actually some fragmentation that would cause seeding and secondary nucleation, right? I don't want to go too much into the continuous crystallization. That would be its own webinar. just very quick, the comparing batch versus continuous. So the batch system is unsteady. So we are always changing some kind of like there is a driving force. So when we're ramping down the temperatures during that temperature ramp, so we have the supersaturation formation, particle formation, crystal growth, all of those things, right? At continuous, at the steady state, we have an equilibrium condition at the same time that the crystal formed, maybe the new one is dissolving, right? So I'm not talking about like that at the molecular level, but at like the process level. So the continuous system is continuous crystallization is always at equilibrium when they are at the steady state. Now when we have this different kind of driving force and this difference in mass transfer, we would have a different crystal habits. And also the particle size distribution would be different. Also the purity would be different. So purity, if you remember my previous slide from the impurity inclusions, when we have less impurity concentration in the solution, we would get less impurity into the crystal lattice. Because in a continuous crystallization, we are adding fresh solvent, fresh solutions. So we kind of adding the makeup and also withdrawing the impurities in the solution. So it's always lower concentration of the impurity inside the crystallizer. Right. So a couple of case studies I have here. So we're going very quick over a couple of those. So before going to the case studies, let me talk through some basic definitions. If you're not coming from pharmaceutical industry or from like a regulatory and have no QA background, so these are some terminologies that I'm going to use during my case studies. So CQA is a critical quality attribute. It could be the form of the crystals, the particle size distribution, purity, whatever it would impact the drug delivery, or the dissolution time, or the bioavailability of the crystals, right? CPP stands for critical process parameter. And so we might have, let's say, 10 different process parameters, but when we run the sensitivity analysis, so we can figure out that from those 10, which are non-critical and which are critical. For example, mixing, as we saw in the previous slide, is one of the critical process parameters, right? But maybe the pressure is not that much critical. So it's just a normal process parameter, it's not critical process parameter. CMA stands for critical material attributes. That's more kind of used in the drug product side, but in a crystallization, so we can make an analogy and say that maybe the impurity at the inlet, the impurity concentration in the starting material in the feed, it could be one of the CMA for our process. PAR stands for process acceptable range. We would have design space that shows that if we change the process or if we change some parameters, how much kind of wiggling room we have, right? And if we go beyond like 10%, so if we are staying like within the plus minus 10%, so we are still getting, so to speak, okay material, so in terms of quality or whatever CQA we have. But if we go beyond 20% or 10%, now we would have significant difference in our product, right? Getting back to my earlier slide, in terms of the scale up, so on the left side you can see kind of a screenshot from my son's math book. He's in an elementary school and they learn about the scale factors, right? So the scale factor here is a dilation. I want to increase the size of the triangle or a shape, and I just times everything by 5 or 10, right? So that's called like a scale factor. And unfortunately, in a pharma, I see a lot of different processes and different companies that they are still using the same approach, right? Now I'm going to a larger vessel, so let's make everything times 10, right? But I showed in the earlier slide that we have this kind of non-linearity of the parameters, that when we go to higher volume, maybe the RPM stays the same, maybe and now the Kurzheim is like 100. So this is very important to have like the good knowledge of the different processes and different parameters and different sensitivities, and understand how these system non-linearities would be important for a scale up. So don't do the elementary school math. So we have to do better and in more intelligent way of the scale up. So in this case, our clients, they developed kind of crystallization recipe in the lab scale, 100 milliliter, 5 years ago when they were running the clinical stage phase 1, 2. And then they did a clinical trial. They submitted the clinical trial data for the regulatory submissions. Everything approved. So they were lucky that they had a product that's getting to the market. Now they want to go to manufacturing. So the process of the process development and the product development and getting all the clinical data takes 5, 6, 10 years, even more in some cases. Now we are going to manufacturing, and our CQA is fixed now because we develop a tablet so we develop some dissolution kind of analysis and drug delivery and the bioavailability based on the particle size, particle shape, and the form that we got from the lab scale. So their problem was that when they go to larger scale manufacturing, they could not make the same size and form and purity of the crystals for all the reasons that we explained before. So and this large scale manufacturing, it is very expensive to run DOE, right? So you never have like 100 kilo of material to just play around, or you don't have like 10 days at the manufacturing scale to run a DOE and find the best conditions and develop some like a kind of design space just by the data driven models, right? It's become very expensive. Material would be expensive, so solvents, GMP time, everything is going to be very expensive. So that was the problem they had, and they came to us at how we can replicate the particles and the crystals and the product that we got from the smaller scale, from the development stage, now into manufacturing. And we help with different kind of like multi-scale predictive simulations, high fidelity simulations, some CFD, some flow sheet modeling, and starting from the fundamentals of the nucleation and the crystal growth and attrition, all the way to the process. Here, the left side is the recipe that developed in the lab. So they kind of again made everything times 10. They maintain the same slope. And now I want to run the same kind of cooling profile in the manufacturing scale and get this particle size distribution that I want. So for this case, they couldn't do seeding. Normally for these cases, we use seeding as a controller strategy. But for some reason that I cannot discuss much, they wanted to avoid seeding for this case. Excuse me. And it never forms, right? So at a larger scale, now when we have this cooling profile, so it gives us different nucleation rate, different growth rate, and they never could get to the particle size distribution they want. So if we did some sort of like a reverse engineering, some reverse calculation, and so kind of calculated the birth of the crystals, relative supersaturation ratio, the supersaturation ratio from the lab scale, to find out that at which rate, at which parameter would get to that particle size distribution. And now that we develop this knowledge, which is the QBD, quality by design, right? The knowledge-based process and product development, then we use that knowledge to now have like a forward calculation for the larger scale, right? So now we put this different kind of like a parametric study, change different RPM, different cooling rate, and find out that which kind of recipe they have to run in the manufacturing scale to get to the same particle size distribution that they had from the lab scale. And if we compare these two, you can see this is the recipe developed in the lab. This is for now they kind of copy it for the larger scale, but now instead of having a sharp ramp, we found out that we have to cool down, form certain number of the nuclei, then give them longer time to grow. So it's like a controlled nucleation and a controlled growth rate numbers and the sizes, so to get the particle size distribution that we wanted, also impurity inclusions. That was the impact of the cooling rate. Also, when we have the mixing, now we compare the mixing here. You can see at the larger scale and the smaller scale, the maximum shear rate for the larger scale was significantly larger. It's like 10 times larger. So we never could run it at the 300 RPM. We just did a simulation to see the trend of the data at that high RPM. So you would have vibration, you would have like mechanical issues, and normally you in the larger scale manufacturing you cannot go above like maybe 200. So most of the companies they run around like 60 or like 100 RPM. The tip velocity is similar. You can see is like 10 times even more. Maybe 100 times larger at the scale up at the manufacturing scale. And now this kind of like impact of mixing also would impact the tip speed and the attrition and many other stuff, right? Another case is for like a continuous reactive crystallization process, right? That was like a dynamic process, and so we wanted to see the impact of different, let's say, disturbances. In this case, we're changing the temperature. For reactive crystallization, you all know is a reaction happening. Let's say two reagents reacting together, and these two are like soluble in the solvent system. The product of the first reaction is a salt, or is a new molecule that has lower solubility in the solvent system. Now the salt, the product of the reaction, gets into supersaturation and precipitating out, right? So that's the crystallization section. So it's always a battle between the reaction and the crystallization. In this case, we wanted to see what would the impact of the temperature, right? So if the temperature goes up by design, maybe the manufacturing people, they want to increase the temperature for some reason, or it would be like a disturbance in the process, what would be the impact on the particle size distribution, and how many residence time distribution they have to wait to get to the steady state. When we're increasing the temperature, so you can see in the reaction rate here, reaction is a kind of classical chemical engineering reaction kinetic. At a higher temperature, we would have higher rates. So we expect that it would form more solute, more product of the reactions. So the supersaturation ratio should go higher. But maybe the solubility curve is like a steeper than the reaction rate curve. Maybe at this new higher temperature, so we would have higher soluble material. So maybe the supersaturation ratio goes down now, right? So now this is the nonlinearity of the system, that at a higher temperature, we don't know the magnitude yet. So you're gonna see in the next slide, the particle size distribution go higher, go lower, or stays the same. Here I'm showing you the screenshots of the simulations from the inlet of the or like the temperature. So this is like a temperature in the jacket, and this is the continuous mean average size particles that we are withdrawing from this crystallizer. This is a larger scale, and it takes like long time to get to the steady state. You can see, and like 26 hours for the residence time to get to the steady state. And this is something that we cannot do by experiment. So this is the way we use the digital twin and the simulations to predict and have some control strategies and understand that why this happens, and how to control that, and what would the impact on the particle size. So I'm going to run the videos. You can see if at the starting time, let me run both. So this is like a steady state. Temperature is not changed yet. So we get the kind of a steady particle size distribution getting out of, or like I would say like mean particle size out of the crystallizer. After the temperature change, and now we can see it kind of like it bounces back and forth to get to a steady state. And now we get to reach a steady state, but we have with a new mean or like average particle size. Temperature change here was over 3 hours, so we get to a steady state in over 3 hours, but it takes 26 hours for the particle size to get to the steady state. Some screenshots I have from like different different times. You can see when the temperature goes up, we would have some maybe nucleation, some dissolution, some other phenomena that if we compare the like location of the peak, it goes back and forth over time to get to the steady state. If you plot this versus time, mean size versus the time when the temperature goes up, so the reaction is slower, so the reaction is not kicked in yet. So we would have more solubility, higher solubility, and some more fine particles kind of dissolved. So now we get to lower particle size distribution. But when the reaction kicks in, because reaction is kind of like, as I mentioned, slower than the solubility or the dissolution of the particles, now we're getting a lot of solute concentration, so the supersaturation ratio goes significantly higher, and now we're getting very large crystals, and it takes a couple of bounces so to get to the steady state versus reaction versus the crystallization or the precipitation to get to the final size. These are some validation data. Our client took a sample during the changes, but when we compare the before and after change, so you can see the particles are larger, and so we have very good kind of confirmation between the simulation and the validation, or like in the actual manufacturing case. Also, the temperature would have impact on the impurity formation and impurity inclusion. So the reaction is not just one single reaction. We also have some side reactions in the system. So in this case, it was a coupling of the product at a higher temperature. So we would have a higher coupling rate. So we're forming more impurity at this higher temperature. And now higher impurity concentration in the solution would cause higher impurity inclusion into the crystal lattice, right? You can see when we go into, and this is kind of like a nonlinear system. If we go like up to plus the 10% or 10 degree or like minus 10 degrees, so we would have like different amount of impurity formation. And now, if you remember from the impurity inclusion or the effective distribution coefficient, when we are also forming crystals faster, when the crystal growth is faster, also there would be more rate of the inclusion by the solvent pockets, or just by the growth getting to the plane of the crystals at a faster rate. And in fact, at the lower temperature, so they had around 2.9% impurity inclusions. At higher temperature, they had around 4.2% impurity getting to the crystal. So it's like less purity for two reasons. I'm repeating myself again here. Because of the formation of the new impurities, or like the faster side reaction rate, and also the faster growth cause more inclusion into the crystals. Another case study was for like a batch crystallization scale up. That was a greenfield project. So we were working on equipment design. This is like 3 years old project. Is already published. We have a publication on this. You can refer to that. I forgot to put the reference here. Apologies. For the greenfield projects, there is a liberty of buying or designing or making a new crystallizer based on optimum design, geometry, and shape of the vessel or impeller or the baffles, right? And in comparing to the brownfield projects that where an equipment exists and we try to retrofit the chemistry or the crystallization to that crystals. So the first case study that I showed, it was like a kind of brownfield, and you saw that we had to kind of retrofit the recipe and the kind of how we are running the chemistry or the crystallization into the existing equipments. Here we had to run, in this case, this is 300 simulations, and starting from different parameters that you would have for the vessels, let's say volume to height ratio, curvature of the bottom, number of the baffles, size of the baffles, location of the impeller, angle of the pitch blade, all of those things that ended up to like 300 literally like DOE cases, and that become like computationally very expensive, but still cheaper and faster than running in the lab. First of all, we don't have those 300 different designs of the equipment in the lab, right? Or it is worse, it's like manufacturing, like a pilot scale. But with these kind of simulations and this kind of like a parametric study, we could find the best conditions. In this case, you can see the shear rate and the heat transfer that gives us the certain particle formation, crystal growth rate, nucleation that we want to get for our particle size distribution that we are expected. So this was the first step. After finding the best condition of the equipment design, so we move to also the population balance simulations. On the center side, you can see like the validation between the lab scale and the pilot scale and experimental data and the simulations, and then we find out the best condition, best design, and then they went and purchased the new equipment. The bottom right shows the 2D population balance for that case. So let me pause somewhere here. The R2 is the width of the crystals. The R1 is the length of the crystal. So this is a 2:1 aspect ratio. And this is a batch, as mentioned, is a batch crystallizer. Over the time, when the time goes by, so the number of the crystals also goes up. So the peak height also goes higher. So we are growing the crystals and also we are making new crystals, right? So this is kind of like the filter data for like a certain class that if you wanted to kind of monitor here for this population balance, but it was like a very difficult 2D population balance that we had to do it based on the mechanistic data and some data driven parameters that we got from the process. That's kind of more details on like the digital twin, and that how we can run multiple different cases. The video on top shows it is like 1,000 times faster, but like over like 1 day, 2 days. So we can run these 300, 400 cases and get the best condition that we want, and it's gonna very rapid and fast and efficient and cheap for this process development and optimization on a scale up. Right? Getting back to the digital twin levels that I had in my earlier slides, as I mentioned, so we had like multiple levels. So we talk about like equipment model and a process model, but now when we develop this knowledge, it also can be used for process control. So it could be like the highest level of the digital twin with the highest complexities, that there would be model predictive control. There would be some models, some mechanistic models that can be used in a process control as well, right? So if something changes, if a disturbances happen, now our models or the digital twin can control the process kind of intelligently. You may say that CFD or the population balance or all of those calculations going to be very time consuming and computationally expensive. It's not possible to put it in the control systems, but now what we can do is it's called like a surrogate modeling. So we're developing like a response factor, or like the kind of developing from the surface, that if you have like multiple parameters, what will the sensitivity between these parameters and the correlations. And when we're developing the surrogate model, now that surrogate model could sit in the control system and use as an MPC, model predictive control. You can see we would have, this is one of the kind of like dashboard that we have at Procegens. If we work on a process control and have projects on this area for that, how we can start from like mechanistic modeling, high fidelity simulations, then get to surrogate models, then go to the control loops. And we would have like the physical plant level, that would have equipments and sensors and HMIs and network of probes. And also on the virtual side, so we would have some models, some kind of surrogate model, and some like intelligent kind of part of the MPC that's going to communicate in two-way, and you would have this kind of MPC. So I would have more webinars on this kind of plant-wide digital twin in future. So stay tuned for the webinar and how we are using the MPC for the process control. Thanks for the attention. If you have any question, feel free to shoot me an email at info@procegens.com, or subscribe to our LinkedIn page, that we put more news about future webinars and a presentation. And thanks for your time.